side A, side B, and they're separated by a membrane here. Embedded in that membrane is a pump, right? A membrane protein called a pump. And that is going to facilitate the movement of particles from side A to side B. Now we start off with equal concentration on both sides, and because there's a higher affinity here on this side of the pump, we're going to have more particles binding onto side A and moving across to side B. Now in this image at the bottom, what we've reached is a point that we call steady state. And steady state means we have movement of particles, but we do not have net movement. We no longer have net movement. So we've moved a certain amount of those particles across side B. We've changed the concentration using that active pump, but then we get to a point, although there's movement continually happening, there is no net, there's no further net accumulation of particles on either side. Okay? And that's what we call steady state. Let's look at some of the characteristics of a pump. They are membrane proteins, right? They are a type of membrane protein. They also have enzymatic action. And as a result, they can harness energy to move molecules against their gradient. They have specific binding sites. So there's a certain level of specificity at their binding site. For example, the sodium potassium ATPase pump has the special ability to bind sodium on one side and potassium on the other side. They also demonstrate saturation. And that means that they can become fully saturated and there's no further movement because you don't have any pumps available. Now there are two types of active transport. There's primary active transport and secondary active transport. And the difference between these two is the source of energy which fuels this movement. So as we said, energy input is required for active transport. And the difference between primary and secondary is the source of that energy. We'll see that for primary active transport, there's ATP hydrolysis involved. Okay. So the energy to move these molecules against their concentration gradient comes from breaking an ATP molecule, using that phosphate and harnessing the energy from that phosphate to move those molecules. So let's look at this scenario here. Okay, again, we have this ATPase pump. This one is specifically called the sodium potassium ATPase pump. And that ATPase indicates to us the enzymatic activity of this pump. Okay, and it's going to bind 3 sodium ions on the intracellular side. And basically, ATP is going to be hydrolyzed. And hydrolysis simply means that we're going to use water to split that third phosphate off. If you remember, adenosine with 3 phosphates attached is what ATP is formed from. And when we hydrolyze that third phosphate group, we can harness that energy in the form of this phosphate. Okay, so this phosphate is going to fuel the energy for this protein to shape shift or change its configuration, and that's going to release those sodiums on the extracellular side. Okay. Now, when the binding site is open to the extracellular side, we're going to bind 2 potassium ions, which are coming from the extracellular space, and we're going to again hydrolyze this phosphate, and that is going to change the shape again such that the binding site is now exposed to the intracellular side of the cell. Okay, and so those 2 potassiums can be brought in. So using ATP, or harnessing energy from hydrolyzing ATP, we can spit 2 sodiums out of the cell and bring 2 potassiums into the cell. And this is an example of primary active transport using the sodium potassium ATPase pump. Okay, we're going to see a lot about that pump as we go on.
So it's 2 sodium out.
So it's 3 sodium out and 2 potassium comes in. All right, and again, that's going to be a very important pump in terms of maintaining the membrane potential, as well as we're going to see that a lot when we talk about cell signaling, how neurons speak to each other, electrical impulses and such. Okay, let's look at secondary active transport. Secondary active transport. Now, energy is going to be released from the diffusion of ions to fuel this type of transport. So we're not going to use ATP. We're going to use the energy that is released when an ion diffuses or moves along its own concentration gradient. It's going to be a result of the previous active transport of this ion. Okay. Let's look at some examples. The first one is the sodium-linked glucose pump. The sodium-linked glucose pump. So what we have here is this pump in purple. We have sodium moving along its own gradient. So sodium is moving along its concentration gradient, and glucose is trying to move against its concentration gradient to get into the cell. And so what we do is we harness the energy of sodium moving freely, and we use that energy to fuel this pump to bring glucose into the cell. Okay, so we're basically capitalizing or taking advantage of an ion that is moving against its concentration gradient to actively pump another molecule that is trying to move against its concentration gradient. Okay, so this is like a 2-for-1 here, 2-for-1 deal. We're bringing one ion in the way it normally wants to, and glucose is sort of hitching a ride using the energy that was released. All right. The second example here is the sodium-linked proton pump, and very similar scenario happening here, just that we're using this in opposite direction. This is called counter-transport as opposed to co-transport. Counter-transport is sometimes called an exchanger, and we'll see that when we look at the muscle system. We'll talk extensively about certain exchangers. So sodium, again, is moving along its concentration gradient, and that energy is being harnessed to fuel this pump to move hydrogen against its concentration gradient. Okay, so hydrogen is being actively transported out of the cell using the energy of sodium. And this is called counter-transport or the exchanger system. All right, any questions so far? So that was active transport, primary and secondary, and we differentiated that the source of energy was what set those two apart. Now this is a very helpful table, very useful, helpful table. This is a great study tool. Okay, I advise you to sort of clear out the boxes of this table and try to recreate it on your own, not from your memory, but from your understanding of differentiating these different types of transport. For example, we know that passive transport always moves down the electrochemical gradient, whereas active transport always moves up the electrochemical gradient. Okay, we talked about the energy source for active transport. Primary uses ATP, whereas secondary is going to use the electrochemical gradient of some other solute. Alrighty, so be able to recreate this table from your understanding as a way to help prep yourself for Monday. Now let's sort of tie all of this together and talk about how active and passive transport are coordinated within the cell. Okay. Now the intracellular and the extracellular compartment have different concentration of certain ions. All right, we know that, for example, sodium is, or the intracellular fluid is very poor in sodium. It's usually about 4 millimoles. The extracellular fluid is very rich in sodium, usually about 140 to 145 millimoles. Potassium is the opposite. The intracellular fluid is rich in potassium, usually about 140, whereas the extracellular fluid is poor in potassium. And so what sets these concentrations is the presence of the pump, the sodium potassium ATPase pump, as well as the fact that we have leaky channels that are moving sodium and potassium into the cell or out of the cell. Let me explain what that means. So we have this sodium potassium ATPase pump, and it's pumping sodium out and bringing potassium in. Okay? And because the concentration of sodium in the cell stays the same, there must be a mechanism by which this can be balanced. And that's because sodium can leak back into the cell. And that keeps the amount of sodium or the sodium concentration in the cell at a steady amount. Okay? Because we have sodium being pumped out, and then it's going to go around the back end and leak right back in. Same thing with potassium. Potassium is being pumped into the cell, and then it's going to leak right back out. And so we have both active and passive transport, really establishing the set concentration of these ions in the extracellular environment and in the intracellular environment.